Hey guys, my name is Max and welcome to Simple Biology. Today we're going to keep talking about Mendelian genetics and we're going to go into some of the more complex ideas and concepts that fall under this category. Now as we go into these concepts, it's going to be a little bit interesting because a lot of these ideas were not actually from Mendel and not even from the time that he was alive. In fact, when Mendel was alive, he came up with just the basic structure of genetics, and from there it has expanded greatly because of his foundation. And that's why we're going to be getting into these things. So first, we're going to kind of fill out our idea of dominant and recessive genes. Now, in reality, some genes show what's called incomplete dominance or codominance. These are two different forms that a gene can take. Now, incomplete dominance is when the two traits mix in some way. Often we see this with like flowers. Now, there are certain flowers that if you breed a white flower and a red flower, you'll get a pink flower. This is incomplete dominance because the dominant trait is not completely dominant over the other trait. Instead, they just kind of mix, they blend. The other form is codominance, like I mentioned, and this is when the two traits are both expressed fully. There's no mixing, but neither one is being subdued. So one good example of this is cows, and they have a couple different forms, but one is black and white spots or brown and white spots. It looks it looks a little bit different with different breeds of cows, but like large spots and patches of color. This coloring pattern is known as roan, and often it results when the parents are two different colors, and they just kind of the offspring just kind of take both colors. The colors don't mix; they don't just blend together, but neither one is absent. Another great example is with your blood type. And now, if you know anything about blood type, you can have A blood type, you can have B blood type, and you can have AB blood type, which is basically both blood types. It's not like a mix between them, but both of them are actually present within your blood. And now that we've kind of defined this, there's an interesting little thing. Almost no genes are actually completely dominant or recessive if you look closely enough. So one example of this would be albinism. Now, if an individual is homozygous recessive for albinism, they will be an albino. They'll be completely lacking in pigment in their skin and in their hair and just throughout their body. The reason for this is because they'll lack a certain enzyme or it'll be dysfunctional, and this enzyme will function in producing that pigment. Now, because they can't produce that pigment, they'll be an albino. But a heterozygous individual will be still lacking some of that pigment. However, one gene is enough to make enough pigment that you won't really be able to tell that they're heterozygous. But at a molecular level, there will still be a difference in just like the amount of the enzyme. Even though it has no difference on the large scale, at the small scale, it would seem sort of like incomplete dominance rather than complete dominance. Now the next concept we have to get into is multiple alleles. Now when I first introduced alleles, I used the example of hair color. I said you can have an allele for black hair color, you can have an allele for brown, for blonde, maybe redhead, whatever it is. I also said that many of the alleles we'll be dealing with only have two alleles. But sometimes this will not be the case. 3 or more forms of a gene can exist, and they do exist in countless occasions. The typical example we will be using is the ABO blood group. Now there are 3 different alleles for this. For when we talk about this blood group, there's the A allele, the B allele, and the O allele. Now O is completely recessive, but A and B are codominant. This is where it gets a little bit messy, but as a result, you can have the blood types AB if you have both A and B. You can have the blood type A if you have either AA or AO. You can have the blood type B if you have BB or BO, and you can have the blood type O if you have double O. The O would be the heterozygous recessive, but because there are multiple alleles, we have to take these special circumstances into account. you can actually still use a Punnett square for this, but you can have more than just homozygous, homozygous dominant, heterozygous, and homozygous recessive, because you could have different combinations of different alleles. Next, we have epistasis. Now, epistasis is when two completely separate genes will affect the same trait, or it can be even more than two, but when there will not be just one gene that will influence the trait. Now, often the case will be that one gene will describe how the trait exists. So, if we have hair color, the one gene will be the gene that describes whether you have brown hair, black hair, or blonde hair. But there will be a separate gene, maybe, that can act as a complete shutoff switch. So if you're homozygous recessive for the other gene, or it doesn't have to be recessive, but often it is, then you will be completely lacking in the first gene. And so this there's an interesting interplay when we have this, and I'm going to give you an example that we're going to work off of. Now, I've been using hair color a lot, and this it actually really is the best example for this. Essentially, you'll have the first gene, and this gene will determine the color of the pigment in your hair. So let's say black, brown, or blonde. Now there's a completely separate gene, which is almost always dominant, and so it very rarely plays an effect. But in a certain population, it might play a large effect, and so this other one will determine whether or not the pigment is actually made. So both of these will affect hair color if they're one way or the other, but in different ways. What ends up happening is that if you're homozygous recessive for the second one, you will be like white-haired. You'll be like no hair color. But if you're homozygous dominant, or if you're heterozygous for the second gene, then the first gene will determine your hair color. So this is our dihybrid cross. It's not color-coded specifically for epistasis, but you can use a little bit of imagination. The yellow and blue squares will be where the second gene that determines whether or not the pigment is actually present. These yellow and blue squares are where that gene is homozygous recessive. So at these four locations, you'll have no hair color. You'll be let's say white-haired, and then you have all these other squares, and there can be other interplays there too, but in any of these other squares, you'll have the hair color that depends on the first gene. So you can work out the different statistics for that specific, for what you're given in a certain situation, but these four squares will be where you have that epistasis, where the second gene will completely eliminate the first gene's effect. You may also notice, in this case, you'll have the homozygous dominant for the first one. These two will be homozygous, or it will be heterozygous for the first one. This will be homozygous recessive for the first gene. If you take these out, in this specific case, it will not affect the outcome of your cross. The different ratios are not actually affected if you take out these four specific squares. It's not always the case, but right now it is, and it can be helpful to know that and to remember that. This last thing we're going to do, and this is polygenic inheritance. Now, essentially, when you have polygenic inheritance, there will be multiple genes that will affect a certain outcome, that will affect a certain trait. This sounds a lot like epistasis, but there's a slight difference, and that is that there is a continuum. So, an example might be skin color. It's not really this simple, but let's say, for example, that human skin color is driven by 4 genes. Okay, and these genes can either all be dominant, all be recessive, or some combination of dominant, recessive, and heterozygous. For simplicity's sake, you can have a certain number. You just add up the number of dominant alleles. And so, in most cases, only the number of dominant alleles total will matter. So all these different genes that will control that skin color will just be piled up into one big pool of genes, and this will be what determines that color. If you have one dominant allele, that'll give you one color. If you got 4, that'll be a different color. 7 will be a different color. 8, 1, 3, just whatever it is. All that will be really important to know is the ratios for these polyhybrid crosses. A polyhybrid cross is when you have more than one hybrid. So in a polyhybrid cross, the two parents are heterozygous for each of those genes that will affect that polygenically inherited gene or whatever trait. And these are the ratios, the ones in the red boxes. You can also use a Punnett square to do this, but it gets really complicated when you have multiple genes. And so just memorize these ratios. If there's only one gene, it's 1 to 2 to 1. We've already gone over this with Punnett squares. This is a monohybrid cross. If you have 2 genes, then it's 1 to 4 to 6 to 4 to 1, and that would be a dihybrid cross, but the ratios are slightly different in polygenic inheritance. You can also have 1 to 6 to 15 to 20 to 15 to 6 to 1, or 1 to 8 to 28 to 56 to 70 to 56 to 28 to 8 to 1. Now, you can memorize these. That's fine. I personally would not. This is what's called Pascal's triangle. If you know how to build this, do it, and every other line will give you one of those ratios. If you don't know how to do this, it's rather simple. You can look it up on any math website, or you can just memorize these. But this is just an easy way to know this if you know how to set this up. It really just involves a little bit of adding and putting ones in different places. So that's really it. That's all that we are going to cover for polygenic inheritance, and for Mendelian genetics as a whole, it's all just as simple as that.